This is our fourth video on biodiversity. In the last video, we were looking at genetic variation, but we're actually looking at genetic variation within communities. So that community variation that lets us sort of determine these different species, who's related to whom, how did we evolve, what ancestors do we share, who are sort of our closest relatives within these different species. However, within a species, you can also look at that genetic variation. And so we looked at morphological variation within a species, but now we'll look at genetics. So we have to look at genes that would change or mutate relatively frequently if we're trying to compare within a species. So genes that aren't very important, or potentially regions between important genes that are sort of semi-conserved. So here we're looking at probably not an actual gene at all, but 6 different people. We've sequenced some region of their DNA, and we're comparing it comparing the sequences to each other. And the SNPs in that gene sequence have been highlighted in red. So we can ask this question: who is most closely related to person 1? And we can answer that question by looking at differences. So it's likely that these different people with the same SNPs as person 1 are more closely related to that person because they have these same differences. And if we're actually looking at genetics, we're going to be looking at many different SNPs because any one SNP, you have 4 options of what it could be, so the probability of mutating to that same option is actually relatively high. So we have to look at many different locations. So here, person 1, if I were trying to figure this out, I would write down what their SNPs were. So it's GCG, because we can block out all the rest of that information because it's all the same, so it doesn't matter to us. So person 2 is TCA, that's 2 differences. Person 3 is ATG, so that's 2 differences. Person 4 is ATA, that's all 3 are different. And then person 5 and 6 are both TTA, right? So TTA is also 3 differences. So of these, 5 and 6, of these 5 people, we can't say whether person number 2 or person number 3 are more closely related because each of them has these 2 differences. So we can't say who's more closely related to person 1, but we can say that person 2 and 3 are more closely related, or more likely more closely related to person 1 than person 3, 5, or 4, 5, or 6. We can also ask the question, who's most closely related to person 6? And as we saw, person 5 shares all 3 of these mutations. So it's the same spot and it's the same mutation. So given that information, it's highly likely that these 2 people are more closely related. The more SNPs that you have to compare and the more different genes that you're looking at, the better you can the better resolution or confidence you can have in those relationships. So whether we could really draw these conclusions with this little data, probably not. But given this tiny exercise, we can say with fairly high confidence that person 5 and 6 are most likely the most closely related of this group. I did want to highlight one other thing that genetic diversity can tell us within a population. When we have diseases that are introduced into an area and we don't know where they came from, what we can do is we can look at populations of that disease in other places. So like right now, we could look at the sequence, the genetic sequence of the COVID virus. And that's how they're trying to figure out where it came from, by comparing that sequence of the COVID virus to COVID viruses found in other animals. And they're also trying to track its movement around the planet based on differences in that sequence between the ones that have infected humans, right? So if you think about how that works, let's say we have some disease X introduced here, and we sequence it, and it has all of the ones that we sequence have really similar genetic sequences to each other because it was likely introduced, because it was introduced here. It is either reproducing asexually, right, and making more of itself, or maybe two were introduced there, or a small population was introduced. Either way, you're going to have limited genetic variation. This likely came from a larger population somewhere else. So one other thing I studied in grad school was Phytophthora ramorum. It's the causal agent of sudden oak death. It was introduced to the Bay Area, but they didn't know where it came from. So one thing you can do is look at other areas of the globe and where there is Phytophthora ramorum, which you could identify based on its genetic sequence. And then you can look and read that genetic sequence and sequence many individuals within that population. And where you find the highest genetic diversity, not that this picture helps anything, that is likely the place where this one came from. And you can see if the sequences present in this population match any or are similar to any in this larger diversity population, because that population has probably been in that area longer, has more individuals that have been interbreeding with each other or exchanging genetic information in some way or mutating over time to differentiate. So we would have more genetic diversity in the place that that pathogen was the longest. Here we have maybe one or two individuals getting introduced into a new area. So that's going to limit their genetic information to whatever was contained in those two individuals. And changes will accumulate over time, but that takes time. So there'll always be less genetic variation in these sort of introduced populations. So you always want to track it back to the highest genetic diversity. Okay, so why is genetic diversity important? Why do we need that as part of our biodiversity consideration? We talked about this a little bit in the extinction lecture, but one of the major things that we'd be concerned with is inbreeding depression. And so here is kind of a picture of what that can look like. So say we have these snakes, and at gene A, there's two different alleles that you can have. You can have a big A, or you can have a little a. The big A allele is going to be, let's say, a functional copy of some gene that allows you to survive and reproduce. Little a is a broken copy of that same gene. So if you have both copies, one functional, one non-functional, this non-functional one doesn't show up in the physical appearance, the phenotype of this snake, because there is a functional copy doing the thing that needs to be done. It looks just the same as this other snake that has two functional copies, because all you need is one. So these two snakes make snake babies, and those snake babies get a random combination of their parents' alleles. This snake gets one copy of each, this snake gets one copy of each, and let's say that this is a very small population that has a restricted habitat, very fragmented. It might be likely that these two siblings end up mating with each other. And just by chance, this offspring ended up inheriting both of these broken copies. So its survival, its ability to survive and reproduce is going to be impacted because it doesn't have this functioning product that it's supposed to make for its normal function. If this snake instead had outbred, so if it bred with a snake that was not closely related, it's less likely that that deleterious allele, that little a, is going to be present. And so if you are outbreeding and you outbreed with somebody who has two functional copies, all of the offspring will get a functional copy from that parent. So it doesn't matter if this parent gives them a broken copy because they'll have this functional copy. And that's the problem with inbreeding, is that you're more likely to have the same broken copy as people you're more closely related to. And that's why it's considered gross to mate with your siblings or mate with your relatives is because your children are more likely to be messed up and have problems, have genetic disabilities that don't allow them to function normally or die at an early age. So that's why people don't mate with their close relatives anymore, but it used to be fairly common practice. And in some like monarchies, they would only reproduce with each other to kind of keep the lineage pure. And then you would start to see these genetic disabilities accumulate in these people. So like hemophilia and like a weird jaw structure, all of these would accumulate over time. So it's unlikely we would get a family that looked like the Lannisters. It's more likely that they would look like Tyrion Lannister as opposed to you know, the other two beautiful ones. So this is a white tiger, and white tigers are white because they have a broken copy of the gene that would normally be making their hair color orange, right? So that orange pigment is broken, so no pigment is produced in those areas and they look white. People like the look of that. They wanted to use them in the circus. It looked kind of like stunning. So people would breed two white tigers together because then there's no functional orange gene. You only have the white gene option. So you're only going to get white tigers. So that's ideal, right? That you don't have to breed tigers and then get a bunch of orange ones you don't want and maybe get one white one. You very successfully get white tigers. But if this happens for an extended period of time, other broken copies of genes tend to build up. So this tiger has problems with its respiratory system and teeth. Like it probably would be a terrible predator out in the wild. Its nose is kind of pinched up here. It has no eye color, right? These kind of like watery white eyes that tend to get cataracts. So many different alleles that can be linked together. So here the white coat gene is linked, so it's very close on a chromosome to a gene that causes the optic nerve, so for your eyes, to be wired to the opposite side of the brain. So if you are breeding tigers for their white coat, you're also breeding tigers to be cross-eyed. And all of these together are going to lower these tigers' quality of life and their ability to survive and reproduce in the environment. So this is no longer allowed to be able to breed tigers for their coats. It's cruel. You can also consider how we get purebred dog breeds. So these two had the same wolf ancestors, right? However, we took those wolves and over time we selected for very specific traits and had completely different looking lineages of dogs. But to get those and to get these really specific looks, you would then breed those dogs with each other so that you would enhance that look in that dog. And that's how we get these really strange animals that look nothing like each other. And you know, if you have Chihuahuas or Boston Terriers, sometimes their heads are too big for their tiny little bodies, so that when a female gets pregnant and gives birth, she can't actually push out her pups. She has to have a cesarean section because their heads are too big. And that comes from our inbreeding of these dogs to get certain traits. So here's our pre-dog population, our wild dogs, and there's variation with that in that population and variation within the offspring. And so we can select for maybe ones that have a temperament that we like better. And interestingly, when I think it was with foxes, it was a terrible experiment. Somebody was trying to domesticate foxes or get a domesticated fox breed. So they would just get foxes and they would kill the ones that were more aggressive. And over time, just letting those less aggressive ones breed together, they tended to get curlier tails and floppier ears because for whatever reason, uprightness and tail curliness were both associated for whatever reason with a lack of aggression. And so often there are these linked traits that kind of get inherited together. Okay, so They have done a lot of experiments and data collection looking at how much or how inbreeding affects litter size and the kind of viability of those offspring, because there was a lot of deaths, a lot of deformations, a lot of genetic diseases. Like I used to want to get one of those Burmese mountain dogs until I found out that they almost all die around the age of 7 or 8 because they get this heritable form of cancer. So if you were to cross those dogs with another dog breed, then it would likely have some resistance to that or have some functional copy of whatever gene breaks in those dogs, right? So I didn't get one of those because I don't want my dog to die at 7 or 8. I want my dog to live to a nice healthy dog age, like 15 or something. So looking at the litter size and the coefficient of inbreeding, so this has to do with how closely related the parents are. I think 100% is direct siblings with this inbreeding coefficient. So the litter size for most of these dogs, if they're not related to each other at all, would be somewhere around 6. The more closely related they get to each other, that drops off to 0. You get no successful offspring in most of these groups if they are, more closely related to each other. What am I trying to say here? However, these two still tended to get at least one pup, even if they were siblings. This is a really sad graph. It's about the percent mortality by 10 days, so puppies that were born but then died within 10 days. And this is based on how closely related beagles were together when they were having children. And so, this is almost 80% mortality of puppies within those first 10 days if they're closely related to each other. So you can imagine how that would function in natural populations. Not that there is some selective artificial process that's happening to get specific traits, but that we just have really small populations, so those organisms are more likely to be related to each other. And we saw this in our extinction lecture when we looked at habitat fragmentation and loss. So anytime you fragment habitat, you fragment a population, and that population is now smaller and it's more vulnerable to random events that are going to decrease genetic variation within that population, and inbreeding, because organisms who are present in that environment are more likely to be related to each other as they're stuck there in that same environment. Both of those lead to a loss of genetic variability, which leads to this reduction in fitness, the ability to survive and reproduce, which that leads to lower reproduction and higher mortality as you start to accumulate diseases and genetic disorders. Both of these result in an even smaller population, so we have this positive feedback loop of decreased genetic variability. And this is called the Extinction Vortex, but inbreeding depression is a contributor and kind of intimately tied to this process, right? Because inbreeding occurs in each of these little spirals. So inbreeding depression is this loss of fitness within a population due to inbreeding. And what type of feedback loop does this look like? It keeps going in the same direction, right? We get the same output from our input. So that's going to take us increasingly to a smaller and smaller and smaller population. So it's a positive feedback loop. So another reason that genetic diversity is important is for that resilience of our populations. So community resilience can be driven by that level of biodiversity of having a high species richness. But within a population, you can also have resilience because of genetic variation. So here we have two different varieties of tomatoes. This Motel variety and the Moneymaker variety have a different response to this particular fungal pathogen. So here's what they look like normally, right? These two on the left were both just given water. The two, so number ones were given water, and number 2 and 3 were both given water with this fungal pathogen in it. And 2 and 3 of the Moneymaker died. They couldn't hack it. 2 and 3 of the Motel variety, which would have a different genetic sequence for some particular or potentially many genes, that would allow them to potentially recognize or be less susceptible in some way to this Fusarium pathogen, right? So they grow not quite as good as normal, but they still can grow to a fairly large size, make tissues that allow them to make food, and probably reproduce. So having genetic diversity within a population will often help keep diseases to a minimum. So if you think about in a large population of individuals, you would hopefully have a lot of variety. If a disease gets introduced into that population, the more similar everybody is genetically, the more likely that disease is to find a susceptible host more quickly. And the more likely that susceptible host is to be surrounded by other susceptible hosts that it can then transmit to. So a disease in a low genetic variety population can travel very quickly and infect a lot of people. That infection curve becomes sort of exponential. And we've seen this with COVID, not because humans are so genetically similar, but because we don't have potentially as much resistance to it. We do, however, have many individuals who are asymptomatic. So they are resistant to the effects, but they're spreaders. So that presents its own problems. However, because we have that genetic variation, we have certain groups of people who, if we had no ability to communicate with each other, to shelter in place, to provide medical care, it wouldn't wipe out the human population because we have some percentage of the human population that's resistant in some way, right? We have variety. So those asymptomatic individuals would then repopulate, right? So if you think about a high diversity population and a disease gets introduced there, it's less likely to initially find a susceptible host. So that's one way that large options for genetic diversity can keep pathogens down. The other option is that when it does get into a host, it's less likely to be moved into another susceptible host because there are fewer in that environment. There's more genetic diversity that is less likely to be susceptible for that particular pathogen. So just spatially, how that disease is going to move around, it needs to be able to move from host to host. And depending on the disease and how it's transmitted, that might be easier for some than others. But in general, having that variety means that pathogens have fewer options for hosts that work for them.